Good evening. It is a tremendous honor and privilege for us. Yes, we're carrying on straight through. Professor Michal Schwartz has agreed to share with us breakthrough, trailblazing research in Alzheimer. I was joking with her and I said that perhaps she'll find personal kind of medication for certain things, but I didn't know that she was going to be receive a Doctor of Philosophy honoris causa from the IDC in June. But Professor Michal Schwartz is going to present to us fascinating things that are truly a source of pride for us, and we're very proud to host you here, and we're very grateful that you agreed to come and talk to us here.
No.
Good evening. I was actually invited to make science more accessible to you, that developed over 20 years in the Weizmann Institute. But when I looked actually at the essence at this conference, I realized that I was somewhat of an alien here. It's not politics or any other topic that is on sort of the agenda when we talk about politics, but Alzheimer truly does worry us and concern us tremendously, and unbelievable, and unfortunately, it is becoming one of the worst diseases, perhaps even an epidemic. There's no cure for Alzheimer, and all those that have been attempted to cure it over the last 20 years have failed. So I wanted to talk to you about our journey that we embarked on to find the source of Alzheimer, but it was a journey that we embarked on. Should I switch into English, she's asking? I can do either or. In Hebrew. Because the presentation is in English. Okay, I'm going to speak in Hebrew. So what have you chosen? Chose Hebrew. Okay, that was a priority. It's a bit embarrassing when there are people who don't understand, but please just tell her perhaps that there is interpreting. They have earphones for simultaneous interpreting. Maybe you could just tell her.
Okay.
Okay, we didn't reach a consensus on the language issue, so. It really doesn't make any difference to me. I can do it in either language. I'm not sure if it's going to be in Hebrew. I apologize for those who have to suffer the interpreting, but if you want it in English as we go along. So my research study dealt over the last 20 years in an attempt to try and decipher the reciprocal ties between the brain and one's immune system. And I'm going to tell you immediately why we dealt with this issue and how a scientific voyage that starts because of simple curiosity, scientific curiosity, can bring about a drug that is based on the understanding of a mechanism and not a medication that is trial and error. As you all know, Alzheimer's disease is in fact an age-oriented disease, but unfortunately there are a plethora of patients wherein there is an early onset of it. And these are cases where it is not an age-centered disease, but rather a genetic one. But with age, the genetic background also has an important role. But the age, unfortunately, is a stronger component here rather than the genetic factor. All of us realize that in the Alzheimer's disease, there are a number of risk factors, inter alia, one of them, the appearance of the accumulation of proteins that have not been removed from, and we can see specific deaths of cells, apoptosis, and we also see a local inflammation or infection. And because of for a number of decades, there were whole research groups that tried to deal it by anti-inflammatory drugs that all failed. And the question is, where was the failure? Was it in the understanding, or was it in the actual medication that was administered? So they all were left with this big question mark. So my research group decided to embark on a journey and to see why in all those degenerative diseases, including Alzheimer, why do these inflammatory medications do not help? So just to remind you that the brain is an isolated fabric of blood vessels, and until we started working about 20 years ago, they saw that the immune cells cannot actually go into the brain, and they interpreted it as if the actual immune system cells had actually entered and pervaded the actual brain, and they need to be eradicated. So the immune system has a very important role in the actual maintenance of the body. First of all, some of them are very ancient roles, and others we've just understood. The main role being that they have to eradicate viruses and bacteria that permeate and penetrate the body and cause infection. And the other thing is to cure injuries, wounds. And the other thing that we've been exposed to more recently is to identify cells that could develop into cancer cells and to eradicate or cure them before they actually turn into cancer cells. The brain actually waived and ceded its right to actually use the immune system, which was, I think, 20 years ago. There was a question asked, a pioneering question. We know that we can transplant lungs, heart, and others, but we can't actually transplant a brain, because whenever it disconnects, any area that disconnects or any cell that disconnects, it basically dies. So we're not going to be able to transplant a brain. So our question in that case was a very simple one. Would it be possible that that kind of brain that is not interchangeable, why does it not actually help itself by using the immune system? So we embarked on a journey and we asked ourselves whether the immune system can't help the brain actually cure itself. And so from that question, it emanated from that, we found a way of actually recruiting and mobilizing the immune system in order to help the brain. It took us 20 years to reach some kind of conclusions and new insights in this science. In 98 and 99, we showed that the cells of the immune system can be needed and can be used, in fact. And we saw that the immune cells can actually truly assist. And in 2013, we saw where the interface actually takes place between the immune system and the brain. In 2015, 2016, we saw that the interface there does not function when people have Alzheimer or at old age. we saw that the mice, the rodents in the lab, don't learn. But when we neutralize the protein that is actually in the interface and it blocks the entrance, can we actually renew the cognitive capabilities of the brain? And we saw that the mouse in the lab that did not learn how to do it, we tried to teach the mouse. And that is the exam that the test that we use. We actually put two objects here, and the mouse learns these objects, and then we change the place of one of those objects, and the mouse starts wandering. And here you can see the slide wherein you can see that the cognitive capabilities of the mouse were actually revived. And an old mouse, 30% of these old mice do have their cognitive capabilities. And here you can see that but there are those who cannot relearn. And here you can see the two red dots. It's where the actual mice got a placebo, neutralizer, and on the other side you can see that they did actually see one, and you can see that there is a return of the cognitive capabilities. So that we then carried on to see what happens with the Alzheimer and whether the interface actually works and whether one can overcome this lack of communication between the immune system and the brain, and that way retrieve those capabilities. So we used model mice of Alzheimer where human genes were actually transplanted into them, that we knew that they were genes that actually bring about Alzheimer, and others can create plaques in the brain. These are mice that were genetically engineered, and here you can see that there are those where they can actually show total Alzheimer when you can see the proteins, you can see also the apoptosis of the cells, and you can see also a deterioration of this communication between the cells as well. But here you can see a mouse that where there's a cognitive deterioration. We use these mice in order to see whether there is a problem with the interface. And here you can see that the protein, the interface protein, you can see the differences between and the comparison between a healthy mouse and those that had been detrimentally affected by Alzheimer, and we examined that and retested that with human brain as well, and we were able to actually see what the problem is. And our question was, why is there deterioration in the actual communication between the immune system and the brain, and how can one overcome that? And in general terms, what we saw, and it is the same with human beings as well, that with age there is a kind of weakening of one's immune system, and one needs to bolster and boost it. And you can either boost it by either just sort of really pressing on them or removing your foot from the brakes. We can either press on the actual sort of accelerator or we can do the other things. Now, why? We decided to remove our foot from the brakes because our immune system that actually learns and remembers is always on the tip of its toes in order to remember things that it hears. So the moment the immune system is weakened with age, it is a function of the increase of that basically brakes that we put on of the immune system. We know that with cancer, the cancer elopes and runs away from the immune system. And we saw that with specific disease, with specific medication, you can actually help and working on that. So we tried to equip the immune system with an antigen in order to remove those obstacles along its path. And that same, exactly the same, the same antigen that basically helped us in cancer as well. So what we do is we put them in a bath of water, and they don't know that in the bath of water there's a platform that is very close to the actual water level, and they learn how to reach it by trial and error to the actual platform there in the bath. And then the mouse can understand how close the platform is to where it wants to reach. A mouse, after it has learned and been treated, it swims directly to the platform. We saw the Alzheimer mice. You can see 3 groups. Those red circles are the Alzheimer mice that even after 4 days of learning, they could not learn how to reach that platform on the water, and they continue hysterically sort of stampeding in the water, and they can't. But here you can see the normative mice that after 4 days, they swim directly to the platform area. They don't even change the velocity. They know exactly where that platform is. So we can see that the mice that we removed those kind of curbing factors and we enhanced the immune system, they learn better than the Alzheimer mice that have not been treated. So here you can see it on the pathological level of these rodents. So here on the left-hand side, you can see the mouse, where you can see the red dots are the protein that was not removed. You can see it in the cortex and in the hippocampus that is responsible for learning and for spatial memory. That same area that teaches us where we put our keys, but doesn't teach us what we need to do with the keys. And that's a difference between an impediment in one's memory and with Alzheimer. As long as we remember where the keys are in the house, our condition is still positive. So here on the left-hand side, you can see big accumulations in the hippocampus, and after treatment, you can actually see that there is an absolute decrease in the bottlenecks and accumulation of these proteins. After we saw that, we have to see how we can implement that and apply that tomorrow. And that is exactly what we did, because in our immune systems, there are proteins that sort of curb things. We call them inhibitory checkpoints, or proteins that inhibit the actual activation of the immune system. And one of them is PD-1, programmed desk protein, that, of course, these antigens were actually produced, and this was the first that was actually approved in the use of cancer patients. So we used this specifically, the same one that was used in cancer. We thought perhaps would those cells actually, if they were used with those antibodies, would they actually would they enable us to remove the barriers with that interface? And then we used others. Here we can see 6 arms, tentacles, and on one of them, when the mouse goes into the maze, so it starts looking in all the arms. So we treated these mice with the same antibody that is used for the cure of cancer, and we checked to see, as a derivative of this treatment, would the mouse be able to reach the platform directly? And you can see that this is a mouse that has been treated. It goes into the maze and swims directly to the platform. In other words, mice who had already lost their memory, we still succeeded in rehabilitating their memory. And therefore, we can see how there is a decrease in the quantity of plaques, but also an improvement in the memory level. So as a result of this cure, in contrary to what was actually directly to the brain tried throughout the world, we actually managed to remove these proteins, and they removed various mutations in the brain. Instead of doing that, we felt that our treatment was not directed to the brain, but rather to the immune system. In order to bolster it, it would be able to overcome Alzheimer in various various onset stages. So we took the model of the Alzheimer, where there is a and be able to suppress it. So we saw that the actual cells actually died because of that. And we said that this actually can help more in situations of dementia, especially if you talk about short-term memory. But in order to examine short-term memory, we did it in a maze that was shaped like a T, and we actually brought the mice into that maze, and that maze only has 2 arms, and they become familiar with them, and the 3rd arm we only open up later.
And then we put the mouse back in the same maze. And if the mouse is healthy and smart, it will check what it's familiar with. And if he's curious enough, he'll go to the new maze. And that is the feature of a healthy mouse. We checked Alzheimer mice, and we could see that every point here is a mouse, and I'd like to emphasize how strong the effect is. On the left side, we have 3 gray columns. The first column shows us that a healthy mouse prefers to arrive at the arm that he's unfamiliar with because he's curious and he remembers where he was before. The red ones are 3 arms into which the Alzheimer a mouse enters. He has no preference. He enters randomly any of them, and this is typical of dementia or Alzheimer, sadly. And when we treated with the drug, which was originally used for cancer, the first 2 columns on the right side, the blue ones, we see 2 concentrations of the antibody. We see emphasis for the new arm with a treated mouse who becomes very similar to a healthy one. In other words, by treating with the antibody, which in the early stages of Alzheimer, we're able to restore cognitive capabilities and also reduce the pathology of the disease. We tested at all stages of the disease. We have a beneficial effect even if we treat before the outbreak of the disease in mice at the height of the disease and even at very advanced stages of the disease. I can't say if that's what will happen in humans, but we see that it is not restricted to the early stages, and that's because we give a boost to the immune system, which teaches it to do what it knows how to do, which it lost with age. We tried to understand why in mice, in a normal mouse that has the immune cells, why they are unable to do the work on their own. And here we cooperated with another group in the Weizmann Institute, which does a number of data analysis. And we found that in Alzheimer mice, the cells are able to work, but they are activated at a very late stage and not at the right time. And we tried to see where these cells are located. We saw that the activated cells in the mouse are next to these accumulations, and we see the same cells in people and in mice, and they're next to one another. We tried to remove them, and they were a low number, and when we gave the treatment, we saw that we would increase the number. So I would like to sum up by saying I apologize once again for the difficulties, and I hope that what was skipped here does not impair your understanding. So I will sum up what we've managed to understand in 20 years. We understand that the immune system maintains the brain. We understood that it constantly boosts the brain, and that dealing with emotional blows and our day-to-day contending with every diversion from any balance requires that a connection between the immune system and the brain, and a failure of the immune system could trigger Alzheimer's. It's not the primary cause, but a failure of the immune system can set the timing of the outbreak. And this treatment it can be if it's very effective in lab rats, and we'll see over time if it works on human beings soon. The difference between what we've done, and we're the first ones in the world to suggest this treatment, and I said without paying a lot of taxes in terms of opposition at the beginning, the difference in what we did, why we think that previous medications failed and why we have a good chance of succeeding. Alzheimer's is very heterogeneous and involves many different factors. Treating one single factor of something that went wrong in the brain is ineffective. We need something that works on all the factors. The immune system knows how to do many different things. So when we enlist it, it can be flexible enough to do whatever is needed. We don't understand everything that the immune system does, but we see the results. The intellectual assets that have emerged from my study and research, a company received a license to develop those assets, and it has a contract very interesting, and it's very promising with a Danish pharmaceutical company, and together with a British company, we're looking to the production or manufacture of a drug that will treat Alzheimer's. The meds for Alzheimer's are a little bit different than other drugs, and the drug regime is very different from the drug regime, for example, for cancer. This work has been supported only by public funding with very competitive grants from the European market. And the people in this room need to understand that the European market is the only source for good research in Israel. I was fortunate to receive 7.5 million euro in total from the European market, and without that money over 10 years, I could not have done what I did. And once again, I couldn't have done it without my outstanding students in Israel and from a number of other European countries, and partners. One of is Ido Amit, with whom I do all the genome work, and a partner in Stanford, and other partners in Europe, and many generations of students, some of whom are already professors in Israel and the world. Thank you.